Hello everyone, I'm Gayatri from First MSc Microbiology, Sacred College, Tiruppur. Let me discuss about theories of antibody protection. What is antibodies? Antibodies are a protein that protect you when an unwanted substance enters your body produced by your immune system. Antibodies bind to these unwanted substances in order to eliminate them from your system. Immune antibodies are classified into five isotypes like IgM, IgD, IgG, IgA, IgE, according to their H chains, which provide each isotope with a distinct characteristics and roles to destroy the disease-causing organisms such as viruses or bacteria or protozoa or some others and block them from infecting human cells. Five theories of antibody protection, which is side chain theory, second one direct template theory, third one indirect template theory, and fourth one natural selection theory, and the fifth one clonal selection theory. Side chain theory. The side chain theory is a theory proposed by Paul Eckledge to explain the immune response in living cells. Paul Eckledge worked on the antitoxins for the diphtheria and their binding to the antibodies in the blood. He hypothesized that antibodies bind to the antigen through the special chemical structures that he called as side chains, v, which is later named as receptors. Paul Eckridge borrowed a concept used by the Emil Fischer in 1894 to explain the interaction between an enzyme and its substrate. Paul Eckridge proposed that antibodies of a receptor to an infectious agent was like fit between a lock and key. Paul Eckridge published the first part of his side chain theory in 1897 and its full form in 1900s. In a lecture he delivered to the Royal Society in London. According to the side chain theory, antibody producing cells, which is B cells, contains a different side chains when antigen encounters the antibody producing cells. It can interact with the side chain and select one of the side chains. It activates antibody producing cells and activated cells then produced antibodies of a selected side chain type. Carl Landsteiner demonstrated that antibodies could be formed not only against antigens but also against various synthetic chemicals, and their theory was abandoned. Direct template theory. Direct template theory was proposed by Bernal and Horowitz in 1930s, Alexander and Mudd in 1932. According to this theory, antigen enters into the antibody forming cells, which is B cells or plasma cells, and direct act as a template for the production of antibody. Since this antibodies is produced from antigen, it is specific for antigen. The antibody specificity is predetermined before the encountering antigen because maturation of B cells occurs in antigen independent phase in bone marrow. Because of this reason, direct template theory was also disproved. Indirect template theory. Burnett and Fenner in 1949 proposed this instructive theory to explain the synthesis of antibody as an adaptive protein. Biochemical functions of proteins by repeated rounds of mutation and selection. According to this indirect theory, antigen enters into the B cell and it binds to the DNA and modified it and forms the modified DNA. Antibodies are produced against antigen and it's also specific for it. But in nature, there is no need for an antigen to enter into B cell and modify DNA for an antibody protection because soluble antigen can activate B cell by binding to its cell surface receptor BCR. Because of this region, this theory was also disproved. Natural selection theory was proposed by Jern in 1955. According to natural selection theory, serum will contains antibodies for all antigens and these antibodies are known as natural antibodies. Antibodies for blood group antigen A and B can given as an sample for this theory. But naturally, serum will not contain secreted antibodies for specific antigen till it enters and activates B cell. Because of this valuable reason, this theory was also rejected. Clonal selection theory. Clonal selection theory is the present day theory which is accepted universally. Clonal selection theory was proposed by Fred McCarthy Burnett in 1957. The clonal selection theory explains the origin of Antibody formation. According to the clonal selection theory, each immunologically competent B cell possesses a receptor for either IgM or IgD that can combine with one antigen. After the binding of the antigen, the B cells is activated to proliferate and form a clone of cells. In the process of antibody protection, antigen selects a single clone from a multiple B cell clone with a different specificity. Thus, this theory named as clonal selection theory. After a clone is selected by the antigen, B cell is activated and proliferated. After proliferation, selected B cell undergoes differentiation to form plasma cell and memory B cells for the specific antigen. Plasma cell secretes antibodies and they participate in antigen removal. The clone selection theory is also practically proved. Thank you for your patience learning. Thank you.